Okay, so today I'll give a quick overview of the single cell RNA-seq exercise that we did last Friday. We'll go through the different sections of the code and I will give a quick explanation of what everything does and why we're using it. And then finally, we will also discuss the two problems at the end. Okay, so the data that we're using today comes from 10x. So 10x Genomics is the biggest supplier of single cell technologies. If you're interested in that, you can click the link here to learn a bit more. They have a couple of publicly available example data sets, and we will be using one of them, which is a data set of the mouse cortex in embryo. So this is E18.5, that's about 3 or 4 days before birth. So the way this works is you have a solution of cells which gets passed through a microfluidics device, which then deposits individual cells into droplets, which contain primers to then amplify the RNA and convert it to cDNA. And these primers include barcodes, which allows you to identify where each cell came from. So each transcript comes from a particular cell, and you can use these barcodes to figure out which cell the barcodes come from. So we will start by loading a couple of packages. So most of these you will have seen before, things like NumPy, Matplotlib. Two new ones that we're using are scikit-learn and SciPy. So SciPy is scientific Python and contains a whole bunch of useful algorithms, and scikit-learn is a machine learning package. And quite the nice thing here is that both of these packages allow you to do very complex things with very simple lines of code. So if you will be doing any more scientific programming in the future, like for instance bioinformatics, you will most likely encounter these two packages again. And gzip is a way of unpacking zipped data. So this is just to unpack compressed files. So we will start by loading the data. In your Google Colab, there will be two cells above here, which allows you to download the data from source to the Google Colab and to attach it to your session. So you can just run those two cells and everything should work. And you will then have access to 3 files. So these files consist of a matrix containing the counts of each gene per cell. So it's the shape on the columns, it will be the number of cells, and on the rows it will be the number of genes. And then we have 2 files describing these 2 dimensions. So we will have a file features which describes the dimension with the genes. So this contains gene names, IDs of the different genes, etc. and barcodes. And these barcodes are the barcodes that identify the different cells. So these are the columns. So we load this data. First we load the matrix. The matrix is by far the largest. And this is a matrix in a sparse format, which we'll go through a little bit later, exactly what that means. And you can load this using a function from SciPy to specifically read files in matrix format. You can see this by the .mtx suffix. And then because it is gz, it will be compressed, which is where we use gzip later on. We then do the same thing for the features and for the barcodes. So what we do with the features is first we make a variable that contains the path to the file, and then we open this file using gzip. So this unzips our file, and we use a function called CSV reader, which reads CSVs into an object. We can then use a sort of loop. So this is a generator expression, which functions the same way as a loop to loop through the different rows here of our file, extracting information. So in this case, the first element in a row will always be the ID, and the first and the second element will always be the name of the gene. And these are the ones that we're extracting here. And we do the same thing for the barcodes, and these barcodes again are the identifiers for the different cells. When we then print a statement describing the shape of the data frame, we can see that we have 1301 columns and 31,000 rows. So that means 31,000 genes and slightly more than 1,000 cells, 1301 cells. So here we have a little bit more explanation of NumPy and SciPy and what you can use it for. You can read this at your own pace. Similarly with the matrices, you have seen the NumPy array before. So this is a very intuitive format, right? Because it's a matrix that consists of, in this case, 4 by 4, and you can pass this as a list of lists with the same dimension, and it will construct for you an array. What is maybe a little bit less intuitive is that this array can also be represented in a different way. Namely, here we have a lot of zero values, and if we have a sparse matrix, that means a matrix where we only save the positive values, or the non-zero values rather, we could describe this in a different way. Namely, we could describe it into locations with values. So we would only have two locations here, 0, 0, and this should be 2, 0, so it's the second row and the 0th column, and these two will both be 1. So this is in fact the very same array as this one, only it is in a much smaller format. And doing this really only makes sense if you have a lot of zeros in your data, like for instance in single cell data. So you can make a comparison. Here we print, for instance, the number of non-zero divided by the whole shape, and you get kind of a fraction of how many values are in fact non-zeros. Okay, now we have our data. The first thing that you're gonna want to do always when you do some form of analysis is to do quality control, right? You want to see if your data has a format that makes sense. And if the quality of the data also is sufficient for the form of analysis that you're wanting to do. So if, for instance, we do a single cell experiment, but most of the cells that we sequenced were dead, then of course it doesn't really make sense to continue with your analysis. And if only some of them are dead, then you might want to remove them, for instance. So a number of good things to look at in this case is the total UMIs per cell. So a UMI is a unique molecular identifier, and this identifies a single transcript. So even if you sequence, for instance, your library very deep, you will get multiple copies of the same UMI, but because this has been amplified, these all come from the very same transcript, so you can then use this to get a better representation of how your data actually looks. But in our case, what we do is we count the total number of UMIs per cell, and this gives you a sense of how many transcripts this cell had. And this is what we often also refer to as library size. Similarly, we will also count the number of cells that express a certain transcript. So this is where these two statements, for instance, are a little bit different. Here we do mod 0 is larger than 0 because we want to know every element that was not 0. And then we take the sum of this. So for instance, if this value would be for a particular transcript 100, that means there were 100 cells that expressed that transcript. So we can plot these values. So we create figure objects and we plot histograms on top of them. And what you can already quite see is that the distribution is maybe a little bit different than what you expected. In many cases, for instance, normal RNA data, bulk RNA data, you will get a sort of normal distribution. Here you can see that most values tend to be 0 or close to 0. So you get these very strongly right-skewed distributions. So this is something that we need to look at, right? We want to discard very low values, but we need to see what it would be a reasonable cutoff. So a way to look at this is to normalize our data. In this case, we will be normalizing these overview statistics, these overview values, and we will do a log10 normalization, which means that we will be looking at the scale rather. So we want to see not if we have 100 or 200 transcripts of a certain kind, we want to know if we have 100 or 1,000. So so we can really compress this very right-skewed distribution into a more compact space, and that way we will get more information about this region. So we do this with a log10 normalization, and we then plot it, and you can see that in fact our data looks quite different now, right? Like when you look at, for instance, the number of transcripts that we have per cell, you can see that there's a very clear kind of normal distribution, that most cells have about 10 to the 4th UMIs. So now you can much clearly set up reasonable thresholds to get the bulk of your data and remove outliers. And somewhat similarly for the transcript, we see also that there is a bit of a distribution here, although there are still quite a lot of genes that are hardly expressed at all. And these we will all be filtering out because these are non-informative genes for our application. And this is exactly what we do here, right? Like we set up here thresholds. So these are then plotted as line on the same plot again. So for the example here, for instance, we are removing every cell that has less than 2,000 UMIs, so that's this line, or more than 100,000, so that's this line. And if you would go back to the first plot, that would mean, for instance, 2,000 would be somewhere around here, and 100,000 would be here. So here it would be very unintuitive to find this threshold, but when you compress your data here, it makes much more sense. And similarly, removing genes that are very low in expression, or at least are not expressed by a lot of cells. And this is exactly what we're doing here. So here we take the total number of UMIs and we're checking if this is larger than the lower threshold and if it's smaller than the upper threshold. And similarly, we are here also doing this for the genes, for the number of cells in which those genes are expressed, and seeing if this is more than the threshold that we set for that one. And here we use these Boolean arrays. So Boolean array means it's an array of true and false statements, so yes or no. And we're removing all the filtered out cells and filtered out genes here. And similarly, we also filter out rows and columns of the data that we are not interested in. And then you can already see we have much fewer genes, much fewer cells, and much fewer genes as well. Now, another thing that is informative to look at, for cells at least, aside from just the number of transcripts that they have, is also how many different genes they express. So this is somewhere that you can, for instance, really use your biological knowledge, right? So for instance, here we use a threshold of 600, so we want to see at least 600 genes expressed by a cell. And if you see less than this, then at least in mammalian cells, that's usually a very bad indicator because there's not a lot of cells that can survive with that few genes expressed. And here if we plot the total UMIs, so the library size versus the number of expressed genes, you can see that this follows a very nice kind of trend that if you have more UMIs, you see also more genes, but it kind of starts here around like 1,000, and it's very nicely correspond to each other. But there is a couple of cells where you might have a lot of UMIs, but these are all captured in the same few genes. And this is often a very good indicator of debris or junk or just low quality sequencing for that part of the data. So this we will also be removing. And here we are doing exactly the same thing again, only we are now removing the thresholded genes, or the cells with very few genes. All right, so now that we have filtered our data, we can normalize it. And one of the big problems that we will have is that we have sources of variation in our data that make it difficult to analyze. So the biggest source of variation in most of these datasets is in fact the library size. So you can imagine that these techniques are very specialized, and you might be capturing more or less RNA from a cell, and this might vary a lot. And if you would do an analysis now, it would be very difficult to see if your results come from the fact that you have different cells in different groups, or if you have some groups with cells that have a lot of transcripts, simply because of differences in the sequencing, and groups that have lower library sizes. So we will need to correct for this. So first of all, we take again the total UMIs, but only for the cells we are in fact using, and we calculate the median. So the median, I guess most of you will know this, but the median is not the same as the mean. It is the most average value. So if you would line up all the values in an array, it would be the middle. That means that if you have, for instance, a couple of really high values, the median will not move so much, whereas the average would be pulled a lot towards those highest values. So this is a reason that many people, for instance, prefer using the median rather than the mean. And in fact, we see that most cells have about 11,000 transcripts. So a library size of about 11,000. So we use this to normalize our data. So we take every column from our data set and divide it by the library size. So for instance, if you have a cell that has 11,000 transcripts, you would divide every value in that column by 11,000 and then multiply it by the median. So that in the end, the sum of every column will be the median value. So all the columns will have been transformed to have the same overall distribution. So it's normalized in that way. And then we will do one more thing, which is that we will downweigh high expressed genes. So high expressed genes tend to drive effects in your data. And it's good to do some form of normalization to filter this out. Because you can expect that if you have, for instance, transcription factors, which will have very low expression values, simply because you don't need that much transcription factors for them to do their purpose. They might be very informative biologically, but if they have very low expression, then it's difficult for the data to pick that up. So we need to downweigh high expressed values, and we can do that with a log transformation. So we do this, and this leads to our normalized data. Okay, now that we have normalized data, we can do many of the more downstream analysis steps. And this part will be perhaps the most complicated part, which is that we will try to go from a very high number of features, in this case genes, to a very low number. And not by throwing genes out necessarily, but by trying to sum similar genes to find sources of variation that are different between cells, but more kind of overarching sources of variation. So you can imagine, for instance, here in this data set, we have developmental data from the mouse cortex. So you would expect to see, for instance, less developed neurons and more developed neurons. So one source of variation that you would expect to see in your data is this maturation of neurons and the different transcripts that will be becoming active or that will become inactive during this process. But similarly, you would also expect to see, for instance, an effect from the capture efficiency between experiments, or an effect from different forms of cell types, so neurons versus astrocytes, for instance. And these are the things that we try to pick up in this dimensional reduction step. Now, first of all, we will be doing is principal component analysis. So principal component analysis is one of the more straightforward ways of capturing variation in your data. And the way to think about this is that imagine your data being plotted, then you would have, for instance, if you would have two dimensions, you could make an XY plot, right? You could have, for instance, if you have two genes, you could plot expression of one gene on the x-axis, expression of another gene on the y-axis. But when you have a very big data set with 20,000 genes, for instance, you cannot do that. So you cannot compare all of this. But many genes will be, in fact, going up and down together because they are part of the same gene module or the same the same biological program. So what you do with PCAs, you find sources of variation. So if you have 50 genes that all go on or off together, they will also correlate together and they will then end up in the same component. So this is what we're doing here. And what we will do first is we will select genes that are changing. So not all genes change. In fact, there are many genes that stay the same in expression between cell types. A good example of this is, for instance, actin, a part of the cytoskeleton, or GAPDH. So these are genes that are very often also used as controls when you do, for instance, a Western blot or a qPCR. So we want to filter those out, and we only want to have genes that vary a lot between cells, so that have very high variance. But for this, we will need to control also for the mean expression, because of course, when a gene has higher mean expression, it will also have higher variance because it will deviate more. And to do this, we will be using a relatively simple machine learning approach called a support vector machine. And here, it's really not important that you know the details that go into this approach. The important thing that you understand here is that we are going to do what we're going to do is to build a very simple model that will try to predict the variance of a gene based on its mean. And we will try to predict this, then we will see if we are right, and we will do it again, and we will do this 20,000 times. Then eventually we will have a very well fitted model that can predict whether or not a gene is more variant than you would expect by its mean expression. So to do this, we use the covariant of variance, so coefficient of variance, sorry, which is usually called the CV, which is the standard deviation divided by the mean. And we fit this to our model. So the model you can just import from scikit-learn, like you can do with many of these things. And we fit our data to it, which we log normalize just to take out some of the depth effects. Then we fit this to a function, and we check how well each gene scores when we use this model, and we can plot this to see how our model did. So here on the x-axis you have the mean expression, and on the y-axis you have the coefficient of variance. So again, this is the standard deviation divided by the mean, and all red dots that lie kind of to the north of this line, these are genes that are more variant than you would expect to see by their mean expression. So these are the features that we will be using. And again, if you don't understand every part of this code, that's all right. This is more just an example. And there are many different ways of doing this. So there are many different approaches, many different programs. This is just one way to do it. And the important thing here, I think, is much more the idea that you think about selecting useful features and what that means for your approach. So we selected here 2,000 genes, and these genes we will be using to build our principal component analysis, or to fit our principal component analysis. And similar to the previous example, you can just import this from scikit-learn. In our case, we select 20 components, and we fit our data. And this is really the only thing that you have to do. And you will then get your components, and you can plot them and see how useful they are. So again, there's also mathematical ways of selecting the number of components. But very often what people do is you simply plot them, and you can see whether or not there is structure in these components. And this is really what you're looking for, right? You are looking for components that are not just capturing noise. You're looking for components that capture some form of structure in your data. And here in the plotted components, you can really see this. So for instance, here you see that this is absolutely not random. There's some kind of gradients going from one way to the other and from here to here. So this is one component, this is the second component, third component, fourth component. But you could combine these randomly with each other. The reason we do in this case from 0 upwards is that every component from the first to the last will have less variance explained. So the first one is the strongest, and then these start capturing less and less variance. And you can see that most of these, they capture quite a bit of variance, and it's around here, so 8 to 10 kind of, where you see that maybe there's not so much structure anymore. It's more like a Gaussian distribution. So you could see this as kind of a normal distribution in 2D. So in normal distribution, you would see this bell curve. And here you see that this kind of also happens with like one region where values all tend to cluster together, and it becomes much more sparse going outwards. And this is usually kind of an indication that you start to capture less useful information here. And you can also see that here. So this is number 8, for instance. And this is the amount of variance that is explained by each of the components. And you can see that it starts to even out. So here we selected 8 components. And this is a matter of choice. So you could also go for 10 or for a little bit less, depending on how much you want your data to be granulated. Okay, so now that we have reduced our data from 30,000 features to about 8, we can think about actually trying to plot all of our data on a 2D plane. So similar to how you would, like in the earlier example, you would have, for instance, one gene and a second gene. What we will now try to do is plot all our cells on a 2D canvas, trying to preserve local structure. So that means that we want cells that are similar to each other to occur in similar regions, and cells that are different from each other to um, occur in different places. And for this, what we will be using is something called a t-SNE. So t-SNE stands for T-stochastic neighbor embedding. And what we're basically doing here is that, and the mathematics behind this are quite complex. We won't go into that too much, but what we're trying to do here is we try to preserve local structures. So between neighbors, neighbors are data points that are most similar to each other, while letting go of the more the data structure a bit further away. So an example of this is the so-called Swiss roll. And the Swiss roll here is a structure of data where there's local cells, local points that are very similar to each other. And then you can kind of fold this. So this is a 3-dimensional structure that in fact has a very strong 2-dimensional structure, right? Because it's actually just a line of points that is curled around. So what the t-SNE does is basically it recognizes this local structure and it gets rid of this 3-dimensional structure. So you simplify your data, but it's all visual. So the t-SNE, for instance, you would not use to do clustering or to do any kind of statistical modeling or ask any statistical questions. It's only a visual way of looking at structure in your data. So again, similar, you can simply import this function from scikit-learn. We will fit it with a PCA structure as we have done before. The random state, because this is a stochastic process, T-stochastic, it requires a random initialization. We simply set this to 1. This is not something you really need to think about. You want to set it to 1 to make sure that your results are relatively stable. If you would take another number, that would work just as well. Only if you then rerun it, it will get to the other result and not to this result. So when you run this, actually your results might look a little bit different than mine. That has to do with the amount of randomization involved. But there should be a kind of local structure similar to this. And again, we only use the components here that we selected before. Okay, so now we have an embedding of our data, which is showing local structures. So we can see, okay, these cells here, they obviously belong together. They are similar in a way. Similar with the cells here, and then here we see kind of a larger structure of cells. And it looks like here there might be a bit of a connection. So it might be that these cells are kind of related in a way. But this is also difficult to say. For instance, if you would have, let's say, this cell here and this cell here, it is impossible to say if they are more similar to each other than, for instance, this cell here and this cell here. Even though this distance is much larger than this distance, it is good to keep in mind that only local structures are maintained. So as far as the t-SNE is concerned, you can compare cells here to the cells in the local neighborhood, so this group here, for instance, but not to any of the other cells here. You simply do not know if they are if they are similar to each other in any way. Okay, so we have a visual representation of our data, but of course we want to take all these cells and assign groups to them, right? Like we have cells here that belong together, but we don't know what kind of cells these are or why they belong together. So the first step there is to kind of cut this into groups. And we will not in fact cut the Disney, but we will simply cut our data, so based on the principal component analysis, and see if these cells in fact are similar to each other and become one group. And this process of separating your data into groups is called clustering. So today we will be using a graph-based approach. There's also many other ways of doing clustering, but currently graph-based approaches are quite popular and they seem to work very well. And the idea here is in a way similar to the Disney, that you use local structures. So you take cells that are very similar to each other and you build a network from these. And then you try to cut this network into subnetworks of highly connected components, called a subgraph. And this is similar also, this is in fact also what spectral clustering is doing. And here you're using eigenvalues, which is not quite the same as a correlation, but it is also related to how similar different points are to each other. So you can initialize this with a set number of clusters. So for instance, now we want to cut our data into 10 groups, and we want to every time consider the 20 cells most similar to one cell, and use this to build the network that we then cut up. So you can run this part of the code and it will give you clusters, and then you can see that in fact there does seem to be similar to what we expected, a big cluster here of cells that are the same, cluster here, and these will be very distinct groups of cells. I think it's very important to note here, for instance, that you still see, for instance, some cells here that maybe belong to a different cluster than the majority of cells here. And I think this is very important to keep in mind, is that it's always good to look at your data from different perspectives. So use different ways of analyzing it to see to what extent it corresponds to each other. So this group, for instance, very nicely separates on the Disney and also very nicely separates in the clustering. So you can be extremely confident that this is one particular group that belongs together. Here there's a little bit more variance, so while this is a very clearly defined cluster, these cells maybe are a little bit less certain, so it's good to keep that in mind when you do analysis, for instance. So now that we have the different clusters and we have this embedding, you can try to figure out what the different clusters are. And in single-cell RNA-seq data or RNA data in general, it's always good to use, for instance, marker genes. So these are genes that we are aware of are very specifically expressed in specific cell types or tissues or processes. And this can help you to define what your different clusters are. So an example here, for instance, is the top row. These are all genes involved in the cell cycle. So TNC and TOP2A are both well-known cell cycle genes, and EOMS is a known gene that is involved in neuroblasts. So these are cells that have left the cell cycle and now are maturing into neurons. So this is a very good indication that in fact what you are seeing here in the data is that we have cycling cells here, and then there are cells that are leaving the cell cycle and are differentiating into adult neurons, and this is the cells that you see on this part. I will leave the rest of these markers to you guys to figure out for the assignment online. But you can do a similar thing as we did before. You can look up these genes, what they represent, and from there figure out what the different clusters are. Okay, so these are this is the tutorial, and up to this point you could basically run all the code and get the results and then try to interpret them. Then there's two exercises here at the bottom which require you to do some thinking for yourself and to fill in the correct parts. So here it's already completely worked out. So when you're following this video, you can basically go through and fill it in like this. But please do try to use your own words to explain exactly what you did in the code. I think this is important. So because it shows that you have understanding of what you're doing, and it also kind of prompts you to think about why you would use certain statements and certain programming um, approaches to solve a problem. So the first thing that we're going to try to do is to create some box plots. So in both problems, we will try to make your data a little bit more insightful. So how can you plot them and get a sense of what is happening in your data? And the first one what we will try to do is we will try to plot get2. So this is one of the genes that we also had here. And instead of plotting it on this embedding, we will try to separate it by a cluster. So for every cluster, we want to know what the get2 expression is. And a good way to do that is to create a box plot. So that the way we're going to do this is that first we're going to create a variable with the name get2, and this we can then easily change. So for instance, if you're interested in a different gene, you can just change it here and then rerun your code. And we will use this later to collect the correct rows from your matrix. So what we do first is we create again a figure object with these statements here, and then we start to create our boxes. So the boxes in this case, not necessarily the boxes that were plotted, we create boxes that contain our data. So first we extract the number of labels that we have. So this is the number of clusters, and this should be 10. Because we did the clustering, we specified we wanted to have 10 separate clusters. So what we can do is we can take all the labels from the clustering object, extract all the unique labels among that, so that should be the values 0 to 9, and then calculate the length. So in total we have 10 values. Then we will create a loop where we loop over this number, so we create a range. So this will be a loop that runs 10 times. And then the list boxes, we will start adding the values of our normalized data. So this is the log10 normalized data. This will be the log normalized data. And we extract only the row where the G name is equal to G, so where the G name is get2, and where the cluster label for that cell corresponds to the label that we are now looking at. So this will give you a list of all the expression of that gene in a particular cluster. So now that we have a list of boxes, we can in fact create our box plots using these boxes, with the labels being the labels that we use to create the boxes. And here you can change a lot of parameters for the look of your data. So for instance, here we're adding every outlier as a plus, but you can also make this whatever. And the patch artist simply makes this a bit more visually nice to see. And then finally, when we have this box plot, we also want to assign color. So if we wouldn't use this part, it would be simply black and white, and adding colors can be useful to distinguish between clusters. So if you keep this consistent, for instance, if you have many figures in a paper, it's always good to have the clusters consistent, so it's easier to interpret between, for instance, figures. So here we do that using a color map. So this is the color map that we imported from matplotlib in the beginning. And for every cluster, we will extract a color from the color map and assign this to the patch artist that we set here. And then we assign these colors to the box plot. And you see that we have two groups that have get2 expression, which is kind of similar also to what we saw here, right? So we have here these cells, and these are also the ones that are active for get2. Okay, then the second part, we are also interested in a little bit more quality control. So one of the things that very often people look at when they do single-cell RNA-seq or RNA-seq in general is the number of mitochondrial transcripts, because this is a very good indicator of cellular stress. If you have a cell that is very damaged or maybe even dead, it will have very high mitochondrial content, because the mitochondria, they will either burst, or you will have a lot of transcripts that have been broken down, but the mitochondria is still intact. So this fraction is known to increase quite a lot in unhealthy cells. So we will try to plot the fraction of transcripts that are mitochondrial versus the total transcripts for that cell. So for this, first you can use this function. So this one is maybe a little bit complicated. And what we do here is we filter using a temporary function. So this one probably you guys have not seen before, which is also why it's defined beforehand. And a lambda function is basically a function you are defining on the go. But it's very much the same as a normal function. So the function here is that we are selecting every gene in gene names that starts with MT, so dash. So this is an indicator for mitochondrial, and then the name of the mitochondrial gene. And we do this for every value of X in gene names. So if a gene has this this prefix, it will be extracted and filtered, and then we have a list of all the names that start with this. Next we go through our list of gene names. And for every value in the gene names, we check if it's in this list of mitochondrial genes. And then we have this Boolean array, so this yes or no list, with every corresponding index of your gene names, that is a mitochondrial gene, will be true, and every other one will be false. So we take our data, unnormalized, just the raw data, and we extract all the rows that are mitochondrial, and we take the sum of that, and then we divide it by the number of total UMIs. So this means that, for instance, if you have a cell in which 100 transcripts come from mitochondrial genes, but it has 10,000 transcripts in total, it will be 0.01. And we can plot this mitochondrial content, and this will give you this plot. And you can see that for most cells, about, let's say, 10% or so, no, less than 10% are mitochondrial. Whereas there are some cells where we have very high amount of mitochondrial genes, and this is usually something that you filter out. Okay, I hope this was informative for you guys. If there are still any questions about any of the things that we did here in the exercise, feel free to email us or ask in the question round that we'll have somewhere next week.